Hello everyone, welcome back to the new session of Biology, Standard 11, Chapter 15, Excretion and Osmoregulation. Last time we studied about excretory organs, osmoregulation. In osmoregulation, we studied about osmoconformers, osmoregulators, euryhaline and stenohaline organisms. Then we studied about excretory organs present in different animals. And then we studied the details of excretory system in human beings. Today we will start with LS of kidney. Before starting the longitudinal section or the details of longitudinal sections of kidney, first we will study the details of all the layers, outermost layers which are present on kidney as covering. Each kidney is externally covered by 3 layers of tissues. The outermost layer is called renal fascia. It is made up of thin layer of fibrous connective tissue. It anchors or holds the kidney to abdominal wall as well as surrounding tissue. Middle layer is made up of fatty tissue called adipose capsule. It protects kidneys by shock absorption. Innermost layer is called a renal capsule. It is a smooth, transparent, fibrous membrane that is continuous with outer layer of ureters. It acts as barrier against spread of infections in kidneys. Now let's start with LS of kidney. LS of kidney shows two distinct regions. They are renal cortex and renal medulla. Renal cortex is outer or peripheral, a red-colored and granular region. It contains malpighian bodies, convoluted tubules, and blood vessels. Renal medulla is inner region with pale red color and striated appearance. It consists of loop of Henle and collecting ducts. All these are arranged in cortical manner to form a renal pyramids. Cortex extends in medulla as columns of Bertini or renal columns between pyramids. Narrow tip of pyramid is called as a renal papilla. There are several pyramids. Renal papilla opens into minor calyx. Minor calyces merge together to form a major calyx. Major calyces unite together to form a renal pelvis. Renal pelvis is a funnel-shaped area. Renal pelvis continues as ureter which leaves kidney through hilus. Structure of nephron. Nephrons are the structural and functional unit of kidney. Each kidney consists of near about 1 million nephrons. Each nephron consists of 4 to 6 centimeter long thin-walled tube that is called renal tubule and a bunch of capillaries called glomerulus. Now, let us study the details of nephron. Nephrons are divided into two parts. One is malpighian body or a renal corpuscle, and second one is a renal tubule. Malpighian body or renal capsule is near about 200 micrometer in diameter, whereas renal tubule is the proximal end which is connected to Malpighian body. That proximal end is wide and blind, whereas the distal end of renal tubule is open, and this tubule is 4 to 6 centimeter long. Now this Malpighian body or renal capsule consists of two parts. First one is glomerulus, which is nothing but a bunch of capillaries or a tuft of capillaries, or you can say the network of capillaries. Second part is Bowman's capsule. Bowman's capsule is a cup-shaped structure into which glomerulus is present. Then coming towards a renal tubule. Renal tubule consists of first part, that is called neck, which is in continuation with Bowman's capsule. This neck then proceeds into PCT, that is proximal convoluted tubule. Convoluted means twisted and tubule means tube-like structure. It is the proximal part which is highly coiled or convoluted, and it is tube-like, so it is called proximal convoluted tubule. This PCT then continues into loop of Henle. This loop of Henle is connected to DCT, that is distal convoluted tubule. It is a distal end, it is highly coiled and it is tube-like. That's why it is called distal convoluted tubule. And this DCT is connected to a short straight tubule called as collecting tubule. Now, the collecting tubule of many nephrons opens together in a common duct, that is called collecting duct, that we'll see in detail further. So these are the total parts present in each nephron: Malpighian body and renal tubule. Malpighian body consists of glomerulus and Bowman's capsule. Bowman's capsule is in continuation with renal tubule and connected to PCT through neck. PCT then continues into loop of Henle. Loop of Henle is connected to DCT. DCT is connected to collecting tubule. LS of kidney contains number of renal pyramids. These pyramids are made up of nephrons. Each kidney contains near about 1 million nephrons. Nephrons are divided into two parts, that is Malpighian body or renal capsule and renal tubule. Malpighian body is 200 micrometer in diameter, whereas renal tubule is 4 to 6 centimeter long. Its proximal end is wide and blind, whereas the distal end is open. Malpighian body is divided into two parts. They are glomerulus and Bowman's capsule. Renal tubule is divided into neck, PCT, that is proximal convoluted tubule. This Bowman's capsule continues into PCT through neck. PCT continues into loop of Henle. Loop of Henle consists of two limbs, that is, descending limb and ascending limb. Ascending limb of loop of Henle continues into DCT. DCT is connected to small straight tubule called collecting tubule. This is then connected to long duct called collecting duct. Now talking about the details of glomerulus. It is a bunch of fine blood capillaries. It lies in cup-shaped Bowman's capsule. A branch of renal artery called afferent arteriole enters the cup-shaped structure and forms a network of several capillaries called glomerulus. Capillary wall is fenestrated, that is, porous. All capillaries reunite to form an afferent arteriole that leaves the cup-shaped cavity. Diameter of afferent arteriole is greater than efferent arteriole. It creates high hydrostatic pressure in glomerulus. It is important for ultrafiltration. Bowman's capsule is a cup-shaped structure. It consists of double walls. Both walls are composed of squamous epithelium. Outer wall is called parietal wall and inner wall is called visceral wall. There is a space in between these two walls called capsular space or urinary space. Visceral wall consists of special type of squamous cells called podocytes. These podocytes are in close contact with glomerulus. That is, the wall of capillaries of glomerulus. There are small slits in between adjacent podocytes called filtration slits. Parietal wall consists of simple squamous epithelial cells. It is thin and continues into neck. The wall of neck is made up of ciliated epithelium. Lumen of neck is called urinary pole. Now this neck leads to PCT. That is, it continues into PCT. PCT is highly coiled part of nephron. It is lined by cuboidal cells with brush border, and these cells are also called as microvilli. It is surrounded by peritubular capillaries, which helps in selective reabsorption. Due to convolutions, filtrate flows slowly and remains in the PCT for longer duration. Now, because of this, maximum amount of useful substances are reabsorbed by blood. Loop of Henle. It is a U-shaped tube that consists of two limbs. One is descending limb and other one is ascending limb. Descending limb is thin-walled and is permeable to water. It is lined with simple squamous epithelium. Ascending limb is thick-walled and is impermeable to water. That is, it does not allow water. It is lined with simple cuboidal epithelium. Whenever I say the word permeable, that means it allows water. And if the word comes impermeable, that means it does not allow water. So, descending limb is permeable to water. That means it allows water to enter inside it. Whereas ascending limb is impermeable to water. That means it does not allow water to enter inside it. Now this loop of Henle is surrounded by capillaries called vasa recta. Its main function is to operate counter current system. Now what is this counter current system? It is a mechanism for osmoregulation. We already studied about osmoregulation. Osmoregulation is nothing but it is the regulation of salt and water balance in the body. So here, whatever the ascending limb is, this ascending limb of a loop of Henle now leads to DCT. DCT is another coiled part of nephron. Its wall consists of simple cuboidal epithelium. It performs tubular secretion or active secretion, in which wastes are taken up from surrounding capillaries and secreted into urine. Now, these surrounding capillaries are nothing but peritubular capillaries which are present around nephron. Now, main function of DCT is that it helps in water reabsorption and regulation of pH of body fluids. Now, next part is collecting tubule. Collecting tubule is a short, straight part of DCT. This collecting tubule reabsorbs water and secretes proton, and it opens into long duct called collecting duct. Many collecting tubules of different nephrons open into a common collecting duct. Types of nephrons. There are two types of nephrons in human kidney. They are cortical nephron and juxtamedullary nephron. Cortical nephrons have shorter loop of Henle which extend very little in medulla. Most of the nephrons are cortical nephrons, whereas few nephrons have longer loop of Henle that runs deep into medulla. These are called juxtamedullary nephrons. Nephrons are richly supplied with blood. About 1/4 of cardiac output is supplied to kidney. Functions of nephron. They are responsible for elimination of waste products and they help in osmoregulation. Blood supplied to kidney. Let's see this flow chart in order to understand the blood supply to kidney. Renal artery collects the blood from heart. It passes blood to the divisions of artery that is called a renal arteriole which enters inside the kidney. Now this renal arteriole then passes the blood to glomerular capillary. This glomerular capillary then passes blood to efferent arteriole. Efferent arteriole is connected to peritubular capillary network present around nephron. So it passes blood to peritubular capillaries. Now from peritubular capillaries, the blood enters inside renal venule. That means these capillaries unite to form venules. So the blood will enter inside a renal venule. From a renal venule, the blood enters into a renal vein, and renal vein removes the blood from kidney, takes out blood from kidney. So blood always enters inside kidney through renal artery and it exits through renal vein. Blood enters into kidney through renal artery. This artery then divides into small branches called renal arteriole. Now this afferent arteriole enters into glomerular capillary. So the blood passes through afferent arteriole into glomerulus. from glomerulus is it exits through efferent arteriole. Now this efferent arteriole passes blood to peritubular capillary network. From this peritubular capillaries, now as these capillaries unite together, they form venule. So the blood from peritubular capillaries now enters into renal venule. These venules unite together to form renal vein. So the blood exits from kidney through renal vein. JG apparatus or juxtaglomerular apparatus. Some smooth muscle cells of the wall of afferent arteriole are modified in such a way that their sarcoplasm appears granular. These cells are called JG cells or juxtaglomerular cells. In each nephron, beginning part of DCT makes contact with the afferent arteriole of same nephron. Cells in the wall of DCT in this region are packed more densely than those in other region of DCT. This is called macula densa. Macula densa and the JG cells together forms a JG apparatus, that is juxtaglomerular apparatus. Function of JG apparatus. It plays an important role in blood pressure regulation within kidney. That means it helps to control the blood pressure. So students, today we studied the details of LS of kidney, structure of nephron, types of nephrons, functions of nephrons, blood supply to kidney, juxtaglomerular apparatus, and function of juxtaglomerular apparatus. I hope you understood the topic. So let's stop here. We'll continue in the next lecture. Till then, take good care of yourselves. Thank you.